Today we're diving into one of the most incredible challenges in all of biology. It's a packing problem on a scale that is just, well, it's almost impossible to wrap your head around. So let's get right into it. Okay, so picture this. I hand you a piece of thread, and it's over a meter long. Your mission, should you choose to accept it, is to pack all of that thread into a space so tiny you'd need a microscope just to see it. It sounds impossible, doesn't it? You'd think the only outcome would be a tangled, completely useless knot. Well, believe it or not, that exact puzzle isn't just some brain teaser. It's a real problem that life figured out billions of years ago. And we're not talking about thread, of course. We're talking about DNA, the blueprint for life itself. If you were to stretch out the DNA from just one tiny little bacterium, you'd be shocked at how long it is compared to the cell that holds it. To put it in perspective, the DNA inside a typical bacterium has to be compacted more than 1,000 times over, just to fit. That's kind of like trying to stuff a kilometer-long rope into a small backpack. So how on earth does it manage this without turning into a chaotic mess?
This is the real mystery we're going to unpack today. You see, it's not just about cramming all that DNA in there. It has to stay perfectly organized, so it's both compact and accessible. So how does life solve this ultimate packing problem? Well, it turns out it's a brilliant two-step process.
All right, so step one is actually something you've probably done yourself. I mean, if you've got a long, messy phone charger cable, what's the first thing you do to tidy it up? You coil it into loops, right? It just makes it more manageable. And that's pretty much exactly what the cell does. The cell takes its gigantic chromosome and organizes it into about 400 to 500 separate, distinct loops. These are called microdomains. It's just like bundling up a bunch of long extension cords. You take one giant mess and turn it into a series of smaller, neater sections. And how do these loops stay put? Well, the cell has these specialized proteins that basically act like little molecular zip ties. They grab onto the DNA, bend it into a loop, and then hold it securely in place. It's a fantastic first step for getting everything organized. So, problem solved, right? We've just bundled all the DNA into hundreds of neat little loops. But you know biology is never quite that simple. This initial plan, it has a huge, and I mean huge, flaw. Let's just do some quick math here. If you were to take one of those nice, tidy DNA loops and measure its diameter if it were a circle, you get this number, 1.1 micrometers. Now keep that number in your head. And here's the kicker. The bacterial cell itself, it's only about 0.5 micrometers wide. So yeah, those loops we just made are more than twice as wide as the cell they're supposed to fit inside. Our neat and tidy loop strategy completely fails. The cell has to get way, way cleverer than that. And this brings us to the second step, which is just pure genius. If just making loops isn't enough, the cell needs to find a way to make the loops themselves smaller. And it does this with a trick that's incredibly simple, but also incredibly powerful. It adds a twist. You know this feeling. If you've ever played with a rubber band, you know what happens. You twist it, and you twist it, and you keep twisting it, and eventually it can't handle the tension anymore, so it coils back on itself, forming a much tighter, more compact shape. That right there, that's called supercoiling, and it is the cell's secret weapon. And the difference it makes is just dramatic. On one side, you've got the looped DNA. It's organized, sure, but it's still way too big. But on the other side, after you apply that super twist, you get the same loops, but now they're coiled up so tightly into a structure that's just massively more compact. This whole process is technically called negative supercoiling because it comes from an underwinding force, and it's the final crucial piece of the puzzle. It takes those oversized loops and shrinks them down so intensely that they can finally and comfortably fit inside the microscopic world of the bacterial cell. So we get what happens, the super twist, but how does it happen? This isn't just some random event. No, the cell has a dedicated, highly specialized piece of molecular machinery whose whole job is to perform this critical task. So let me introduce you to DNA gyrase. This incredible enzyme is the master of the super twist. It's a genuine molecular machine, and the way it works is so precise, so elegant, it's really like watching a magician perform an impossible rope trick. Okay, so let's break down this magic trick. First, the machine grabs one part of the DNA rope and it wraps it around itself. Then it grabs a second part of the DNA rope that's nearby. Now, here comes the mind-blowing part. It actually makes a clean cut right through the first rope, holds the two ends open, passes the second rope straight through the gap, and then perfectly seals the break like it was never there. It's an incredible bit of molecular sleight of hand. And what's the result of this amazing performance? With every single one of those cycles, DNA gyrase adds 2 powerful negative supercoils into the DNA loop. It just keeps doing this over and over again, twisting and tightening until the entire chromosome is perfectly compacted. So the DNA fits. Great. But like we said, this was never just about storage. The way the DNA is packed is actually critical for the everyday life of the cell. It's this beautiful, elegant solution that solves 2 completely opposite problems at the same time. The real genius of this system is how it pulls this off. First, obviously, you get extreme compaction. The chromosome fits. But that negative supercoiling also stores tension in the DNA, kind of like a wound-up spring. And that tension actually makes it easier for the cell to pop the 2 DNA strands apart when it needs to access the information inside. And separating those strands, well, that's essential for pretty much everything a cell does, from reading a gene to making a full copy of its DNA before it divides. So at the end of the day, the cell doesn't just jam its DNA into a box and hope for the best. It creates this perfectly organized, dynamic, and totally accessible library of genetic information. This packing method makes sure that any gene, anywhere, can be found and read quickly, even in this incredibly crowded space. It's the ultimate solution for high-density, high-access storage. And this whole incredible process, this is happening inside trillions upon trillions of cells right now, all around us and inside of us, every single second. It really makes you wonder, right? What other amazing molecular machines are out there, hiding in plain sight, just quietly making all of life possible?